So, what if I told you that the code for athletic greatness, for what makes a champion, is literally written into our DNA? Today, we're going to peel back the layers and look at that genetic blueprint, and then dive into the incredible, mind bending science that's trying to rewrite that code completely.
It's the age-old question, right? Is it all about the thousands of hours in the gym, the grit, the sheer willpower? Or is there a secret genetic lottery that some people just win at birth, giving them an almost unfair advantage? We've argued about this for decades, but now science is starting to give us some shockingly specific answers.
All right, let's get into it. Before we can even talk about editing our biology, we first have to understand the basic software we're all running. And it turns out, our genes can seriously nudge us in one direction or another, predisposing us to certain kinds of athletic performance. Now, if you boil it all down, athletes tend to fall into one of two major genetic profiles. Scientists call these phenotypes, which is just a fancy word for the traits you can actually see. On one side, you've got the endurance folks. Think marathon runners or cyclists. They are genetically wired for insane fatigue resistance and super efficient energy use. And on the other side, you've got the strength and power athletes, your sprinters, your weightlifters, built for pure explosive, high-speed power. So what's actually going on under the hood? Well, researchers have pinpointed a bunch of key genes that act like our built-in athletic software. You've got genes like ACE and PPAR that control how our bodies manage energy. Then you have the famous ACTN3 gene, which helps determine what kind of muscle fibers you have. And there are even genes like COL5A1 that affect how your tendons are built, which can play a huge role in your risk for injury. It's all in the code. And this is where it gets really, really specific. Let's take that ACTN3 gene I just mentioned. It's often called the sprint gene. It makes a protein that's only found in our fast-twitch muscle fibers, the ones that give you explosive power. Well, it turns out there's a specific version of this gene called the RR genotype that shows up way, way more often in elite sprinters than it does in the rest of us. It's one of the biggest clues we have for the whole born not made side of the argument. Okay, but what if you didn't have to rely on the genetic lottery? What if you could just bypass it? This is where the science takes a leap into the extraordinary. Researchers have zeroed in on a single biological master switch, a protein that basically acts as the body's parking brake on muscle growth, and the race is on to figure out how to release that brake. And here it is. Meet myostatin. You can think of it as the body's bouncer. Its entire job is to stand at the door of your muscles and say, "Nope, you're big enough, we're closed." It's a negative regulator, constantly sending out that signal to stop growing. If you didn't have myostatin, your muscles would, in theory, just keep getting bigger and bigger. So how does this parking brake actually work? Well, it's not just a simple on-off switch. First, myostatin is made in an inactive form. It then floats around in your blood in this kind of sleeping state. It's only when a specific enzyme comes along and wakes it up that it can deliver that stop growing signal. And this multi-step process is absolutely crucial because it gives scientists a few different places where they can try to jump in and disrupt the whole thing. And when they do manage to block it, well, the results are just jaw-dropping. In animals that have natural mutations or in lab experiments where scientists shut down myostatin, the effect is just, whoa. We're talking about these mighty mice and Belgian blue cattle that have literally double the muscle mass of their normal counterparts. This was the proof. If you can block myostatin, you can unlock almost unbelievable muscle growth. As you can imagine, this discovery kicked off a massive scientific race to develop drugs that could hack this system. And three main strategies popped up. First, you could use antibodies that hunt down myostatin and disable it. Second, you could create these things called ligand traps. Think of them like little sponges that soak up all the myostatin before it can ever reach your muscles. And third, you could try using the body's own natural myostatin blockers, like a protein called follistatin. So this sounded like a complete game changer, a miracle drug. Not just for building athletes, but for helping people with terrible muscle-wasting diseases. There was just one problem. Taking this amazing science from the lab and making it work safely in humans, well, that's where the story gets really complicated. And you can see that complication right here. Just look at the track record in human trials. A drug for muscular dystrophy, it just didn't work. Another one for a different muscle disease, it failed to help people walk better. And then there's ASO31, which was stopped cold because of serious side effects. The dream just kept crashing into reality. And then there's this one really weird, unexpected success in treating anemia, which nobody saw coming. So what was going so wrong? The biggest problem was specificity. See, myostatin belongs to this big family of proteins that all look kind of similar, and they all have different important jobs in the body. The issue was that these new drugs were a bit clumsy. They were like a key that not only unlocked the myostatin door, but a bunch of other doors you really didn't want to open.
And opening those wrong doors is where the real dangers, the off-target effects, started to show up. For instance, one drug accidentally blocked proteins that are vital for keeping your blood vessels healthy, which led to bleeding issues. Other inhibitors blocked a close cousin of myostatins that's actually important for bone strength. So, you get weaker bones. It was a classic case of the supposed cure causing a whole new set of problems. But here's the thing. Despite all these setbacks, the science is marching forward. We are getting better and closer to being able to precisely edit our own biology. And that forces us to look past the technical hurdles and start asking some really profound ethical questions.
As one of our sources puts it so well, this kind of technology demands our constant attention. We're not just fixing things anymore. We're stepping into a world where we can fundamentally change what it means to be human. And that's a power that we have to evaluate very, very carefully.
And this really gets to the heart of the dilemma. It's the two sides of the same coin. On one side, you have therapy, using this tech to treat horrific diseases or to help someone recover from a catastrophic injury. That's a goal almost anyone can get behind. But on the other side, you have enhancement, using that exact same technology to create super athletes or even to reverse the natural effects of aging. Make no mistake, this is not science fiction anymore. The tools are being built as we speak. So the final question isn't really about what's possible scientifically, but about what we decide is acceptable as a society. Once we can truly rewrite the code for human strength, who gets to use the pen? And where on earth do we draw the line?